the scientific study of life. Density, a dynamic perspective. Another sampling technique commonly used to estimate wildlife populations is the marker capture method. The researchers place traps within the boundaries of the population under study. Captured animals are marked with tags, colors, bands, or spots of dye, and then released after a few days or weeks, enough time for the marked individuals to mix with unmarked members of the population. Traps are set again. This second capture yields both marked and unmarked individuals. From these data, researchers can estimate the total number of individuals in the population. Note that the mark recapture method assumes that each marked individual has the same probability of being trapped as each unmarked individual. This is not always a safe assumption because an animal that has been trapped once may be wary of traps in the future. Density is not a static property of a population but is um, rather the result of a dynamic interplay between processes that add individuals to a population and those that remove individuals from it. Notice this figure. Additions to populations occur through birth, which we will define here to include all forms of reproduction, and immigration, the influx of new individuals from other areas. The factors that remove individuals from a population are death, mortality, and immigration, the movement of individuals out of a population. While birth and death rates have obvious influences on the density of all populations, immigration and immigration um, can also make important contributions to the density of local populations. For instance, long-term studies of belongs ground squirrels, spermophilus breeding in the vicinity of Tioga Pass in the mountains of California show that some of the squirrels move nearly 2 kilometers from where they are born, making them immigrant to other populations. Both Sherman and Martin Morton at that time at Cornell University and Occidental College respectively estimated that immigrants made up 1 to 8% of the males and 0.7 to 6% of the females in the study population. While these may seem like small percentages over time, such rates represents biological and significant exchange between populations. Patterns of dispersion within a population's geographic range, local densities may be or may vary substantially. Variations in local density are among the most important characteristics that a population ecologist might study, since they provide insight into the environmental associations and social interactions of individuals in the population. Environmental differences, even at a local level, contribute to variation in population density. Some habitat patches are simply more suitable for a species than are others. Social interactions between members of the population, which may maintain patterns of species between individuals, can also contribute to variation in population density. The most common pattern of dispersion is clumped, with the individuals aggregated in patches. Plants or fungi are often clumped where soil conditions and other environmental factors favor germination and growth. For example, mushrooms may be clumped on a rooting log. Many animals spend much of their time in a particular microenvironment that satisfies their requirements. Forests, insects, and salamanders, for instance, are frequently clumped under logs, where the humidity tends to be higher than in more exposed areas. Clumping of animals may also be associated with mating behavior. For example, Mayflies often swarm in great numbers of behavior that increases mating chances of these insects, which survive only a day or two as reproductive adults. Group living may also increase the effectiveness of certain predators. For example, a wolf pack is more likely than a single wolf to subdue a large prey animal such as a moose. A uniform or evenly spaced pattern of dispersion may result from direct interactions between individuals in a population. For example, some plants secrete chemicals that inhibit the germinate and growth of nearby individuals that could compete for resources. Animals often exhibit uniform dispersion as a result of antagonistic social interactions such as territoriality, the defense of abounded physical space against encouragement by other individuals, as shown in this figure. B. Notice here uniform bears nesting on small islands such as these king penguins on South Georgia Island in the South Atlantic Ocean. Uniform patterns are not as common in populations as clumped patterns. Random dispersion, unpredictable spacing occurs in the absence of strong attractions or repulsions among individuals of a population, or where key physical or chemical factors are relatively homogeneous across the study area. The position of each individual is independent of other individuals. For example, plants established by wind-blown seeds, such as dandelions, are sometimes randomly distributed in a fairly constant habitat, as shown this figure. See, random. Notice that random patterns are not as common in nature as one might expect. Most populations show at least a tendency toward a clump distribution. Demography, the factors that influence population density and dispersion patterns, ecological needs of a species, structure of the environment, and interactions between individuals within the population also influence other characteristics of population. Demography is the study of the vital statistics of populations and how they change over time. Of particular interest of to demographers are birth rates and how they vary among individuals, specifically among females, and death rates. A useful way of summarizing some of the vital statistics of a population is with a life table. Life tables about a century ago, when life insurance first became available, insurance companies indeed needed to estimate how long on average, as shown in this video, on average individuals of a given age could be expected to live. To do this, demographers developed life tables, age-specific summarize of the survival pattern of a population. Population ecologists adapted this approach to the study of non-human populations and developed a quantitative demography as a branch of population ecology. The best way to construct a life table is to follow the fate of a cohort, a group of individuals of the same age from birth until all are dead. To build the life table, we need to determine the number of individuals that die in each age group and calculate the proportion of the cohort surviving from one age to the next. Cohort life tables are difficult to construct from wild animals and plants and are available for only a limited number of species. Sherman and Morton's studies of the Togo Pass buildings ground squirrels resulted in the life table. The table, this as shown this figure, For instance, or the table reveals many things about the population. For instance, the third and eighth columns list in respectively the proportions of females and males in the cohort that are still alive at each age. A comparison of the 5th and 10th columns reveals that males have higher death rates than females, as shown here. Notice also survivorship curves. A graphic way of representing the data in a live table is a survivorship curve, a plot of the proportion of numbers in a cohort still alive at each age. Let's use the data for building the ground squirrels here, as in this table shown. To construct a survivorship curve for this population, generally a survivorship curve is constructed beginning with a cohort of 1,000 individuals from a population. We can do this for the building's ground squirrel population by multiplying the proportion alive at the start of each year, third and eighth columns as shown here. Number alive at the start of each year plotting. These numbers